Okay. Did you see? Did you guys see the my share the file right now? Okay, good. Give me a second. Let me close my door. Okay, I'm gonna spend near 2 hours to go over the lecture immunology. I mean immune system. And okay, actually, we are going to. I'm going to introduce some key items in this chapter. For example, innate immunity. What is innate immunity? What's adaptive immunity? What's a component of these two different cells or members? Okay, and then I'm going to go over what's the leukocyte adhesion cascade. Then what? Then I will introduce what kind of components uh, triggering in the during the inflammation. And the fifth part is allergy. Okay, what kind of cells, what kind of antibody mediate the allergy. And number six, going to show introduction about the autoimmune disease. Okay, and number seven, I'm going to go over a little bit my experience in bone marrow transplantation. Okay, regarding the hematopoietic stem cell trafficking through the bone marrow, through the peripheral blood. And number eight is very hot topic recently for treatment of cancer patient with a cancer in the checkpoint inhibitors. And I'm going to introduce one set of immunocheckpoint. And I would like to ask you guys, are you familiar with those things actually in this content? Do you have any idea regarding innate or adaptive? Can you distinguish them? Tell them the difference? Do you have any idea what is inflammation? If you don't, that's fine. But after lecture, I hopefully you guys can learn this and turn to your basic knowledge. Okay, I don't know what's your background, but whatever. After this lecture, I hope everyone can get a little bit idea regarding those keywords here in this content. Okay, what is the immune system? Which is just like our military defense system with the Air Force base, right? And army camp, something like that. And also a lot of weapon, tank, missile, machine gun, okay, fighter jet, okay, a lot of different weapon, and also secure coast guard, okay, even the policeman also, MP also, okay. For that's our immune system is our military. Um, it is a military defense system in human being, our body. Try to get rid of the invading pathogen or microorganisms and set up in our body. Okay, and not only pathogens, also a lot of chemicals, toxin substance, and also some kind of allergen, pollen, dust mites also. Okay, the immune system try to resolve those kind of problems. Okay, and those weapons in our immune system, including physical barriers. What's that? Like our skin. That's the first line. Okay, for example. And second is white blood cells. And you are going to learn how many types of white blood cells in our blood. Okay. And third one is antibodies, which release from plasma cells, which is mature B cells. Okay. And number four, we are going to learn another thing is complement. Okay, what it is complement? Did you know that before the lecture? And number five is a lymphatic system. What's this? Okay. And number six, they are going to tell the organs for maturation of B cells or differentiate into different type of white cells in bone marrow. Okay. A lot of hematopoietic stem cells live in there. And number seven, in the end, you are going to learn what's allergy or something like that. Okay. Let's see this picture here. That's the pathogen, okay, including parasites, protozoa, the worm and fungi of fungi or fungus or prokaryote cells like bacteria and even virus. Another thing, prion. Do you know what is a prion? It's kind of protein. It's mutated protein, which it can cause disease in in human, cause it's new make your neuron dysfunction, which is like a mad cow disease. Okay, and we got the military system, right? We got a missile, we got a weapon. So how do we distinguish this kind of cell, this kind of members is not belong, it's not ally, it's not our friends, it's enemies? How can you tell? By those important markers. Everybody got to know this one. We call them this set of protein. We call them human leukocyte antigen. The short name is HLA, which is identical to most elements found in the animals. We call them MHC. Okay, they are basically the same thing. I'm going to ask you a question for you all. Can we transplant or we can do blood transfusion, right? We can receive the blood from the donor, right? In case we got a surgery or injury, we need the blood. We can transplant platelets, right? Can we transplant or receive the white cells from donor? No, right? Because we don't have not exactly identical, HLA components. So they will rejection, will result in a lot of severe consequences, inflammation. So how come red cells and pellets we can do the transfusion, blood transfusion? Because cells without the nucleus, they are not going to present MHC or HLA, those personal markers on their surface. Okay, unless you can find identical twin, then you possibly get a very high chance to get transplantation or transfusion. So therefore, most the cells with nucleus, it must present HLA. It makes it very difficult to do transplantation, even organ or bone marrow. Okay, that's just clarify one thing. And this slide to tell one thing: every kind of cells, epithelial cells, endothelial cells, white cells, they will present their unique markers, ID, on their cell surface. And these markers, we call them HLA, or you can call them MHC. Okay, and MHC is a very important substance, not only the markers for person, but also it can be a receptor antigen. Okay, later on we are going to learn it. There are two types: MHC class 1 and MHC class 2. Okay, and let's look at this diagram to show you what kind of components in our immune system. The first one I mentioned already, hard stuff, barrier, like a compound. Okay, for example, our skin, the mucous membrane in our mouth, trachea, right? And also stomach acid. Okay, those things, or lysozyme in our tears, those things can protect uh, the microorganism or pathogen, set up the root, get infection into our skin or into our cells. That's the first defense system, barrier. And also we can use another different way, physical way. For example, coughing, sneezing, okay, and get a lot of stuff, or vomiting or diarrhea, something like that. That's a physical movement to carry the chance for invading or passaging into our body. And second line is innate immunity. What does it mean, innate? It won't change with the time. They always have the same capability, identical capability. They don't have a memorization. They can memorize everything. This enemy came here before or not, they can. Okay. And they including couple different type of subcell leukocytes, for example, neutrophils, macrophage, eosin, and a basal, okay, and a monocytes. Besides that, they also has another set of protein which produce is excrete from the liver. This kind of protein we call them complement system, which another set of proteins. It's structural protein and also protease. Okay, in the end, they will equip into a very unique structure. Okay, they will drill a hole on the cell wall, cell membrane, and then weapon complement. Moreover, they do have another different activity also. I will mention later. And third part here is adaptive immunity. There are two types basically. The majority of T cells and B cells, and another two type of cells, but the percentage relatively very low. Okay, T cells. There are two kind of T cells, and I think all you should know that the first one is T helper cells. Another one is cytotoxic T cells. T helper cells, he imposed a lot of different functions onto different leukocytes, including adaptive and innate immunity. Okay, we will learn it later, of course. The next one is B cells. B cell can develop after stimulation and will mature into plasma cells. The most important part is generating what? Antibody. Antibody like a missile, which can neutralize toxic substance of passage, leading to phagocytosis and clearance. Okay, from our tissue injury tissue. Okay, let's go next. Let's go over the innate immunity again. Here's the innate immunity. I just mentioned there are several types of different cells: basal, eosin, neutrophils, and the dendritic cell macrophage. And they don't have monocyte here. I want to clarify one thing. The monocytes in the bloodstream, we call them monocytes. After they're activated, they are going to migrate, cross over the endothelial cells into the tissue. The activated form macrophage, okay? And the neutrophils, the most abundant cells is neutrophils in our blood, okay? NK cells, which is innate immunity, which is very useful, like immunity cells can kill the cells got infection. Okay, any passage cytoplasma already. It doesn't belong to the adaptive immunity. And also, it is very useful for treatment of cancer. Okay, it's specific for the cancer. Another one, dendritic cells. Cell is interesting cell, which is like a monocyte, like a macrophage, but it doesn't stay in the blood. It stay in the tissue and the organ. It's a very long lifespan, maybe stay a couple months, still alive. This guy, dendritic cells or DC in the macrophage, they also is an antigen presenting cells. Why is the antigen presenting cells? Later on, I'm going to explain it to you guys later on. Okay. Let's see innate immunity here. You look at that basal, neutrophils, eosin. You close look the cytoplasm inside. a lot of dots here appear with a different color. Okay, if we use a dye like a xylosine and an eosine to label these cells, you can see that the granule appear different color. If they did not change the colors at the staining with the dye, we call them neutrophils. Okay, that's the neutrophils. It appear blue color or purple, we call them basal. It turned to the red or orange, we call them eosin. Using this methodology to classify three types of different subsets of leukocytes, because there are a lot of granules in their cytoplasm. That's we call them granulocytes, and we also call them PMNS. Okay. The other one you look at here, monocytes. Big nucleus, size is much bigger. Okay, and they don't have the granule in the cytoplasm. That's a monocyte. So it's not the granulocyte. So it's a granulocyte. Okay, this monocyte is not activated form, and this activated form we call it macrophage. Another set of, the white cells here is a lymphocyte. You can see the size significantly smaller compared to other types of cells, right? Another thing is what? A very small volume of the cytoplasm, most than nucleus. So you can see lymphocytes, even including T cells, B cells, small size, small volume cytoplasm, big nucleus. Because what they need to activate it, produce a lot of protein, a lot of antibodies, something like that. Okay, so their DNA activity is much stronger. Any questions so far? Can you follow it? If you could, I'm going to continue next slide. Okay. Innate immunity, they all has three kinds of receptor. Toll-like receptor here, not like a receptor, IRIs or IIGI like a receptor. This kind of three receptor can help the root white cells to recognize a substance secrete from the bacteria or pathogen or their structural protein. For example, in bacteria, the component cells or cell wall, LPS, and a lot of glycolipids, lipoprotein, they all components of cell membrane and cell wall. So these three kind of receptors, they can recognize them and bind into them, then generate signal transduction pathway, which eventually will turn on some kind of gene, release cytokine, chemokine, growth factor, which can destroy pathogen or digesting pathogen or activate another different set of white cells. For example, macrophage to execute phagocytosis to engulf the bacteria, and also can release different cytokine or growth factor for healing. Okay, so immune system not like only like a military defense system. It also has a function to do healing process. So don't worry, don't forget this part. Okay, and also beside the glycoprotein like LPS, and also a particular receptor, for example, not like IgI like a structure. This kind of receptor, they can bind to RNA or DNA. Why? Because most of the virus, they need to invade into the cytoplasm and take over the productory machine in the host cells to produce their protein, their co-protein, their DNA. So in those receptors are expressed in the cytoplasm, which can recognize viral DNA or RNA and the triggering activation of those white cells. Okay, and these white cells, including macrophage, dendritic cell, NK cell, and neutrophils. Okay, they all express these three kind of receptors. And the toll-like receptor, a lot of different isoform. Each isoform has their unique binding substance, okay, or ligands. Okay, you don't need to memorize, but you gotta know that. Okay, and they will trigger signaling, induce this very important transcription factor, NF-kappa B. This one is very important, can trigger a lot of inflammation reaction. I mean, a lot of activation of immunocytes. Okay, turn on a lot of gene related to cytokine and chemokine. And toll-like receptor, there are three kind. These three isoform 739. This one is taking care of the virus DNA, virus genome, RNA, DNA after the invasion. So those kind of receptor, toll-like receptor, which receive the signaling, the enemy is invading our country. So we are going to react in activation our weapon, our cells, and tell another different type of innate immunity or adaptive immunity, try to help me out, kill me, sacrifice me, okay, because the virus in my body already. Or recruit a lot of different white cells around the injury site, damages tissue to kill them, uh, uh, clean all them, and then later on we are going to do healing process. Okay, so that's important, very important toll-like receptor. Second one that you mentioned, non-like receptor. Which one? This receptor is not unusual. Okay, I try to say not like a toll-like receptor. They don't have a really receptor. They use the pole, they use the channel. Okay, and sometimes they use the phagosome, which engulf the particles in there. So they actually don't really has a transmembrane receptor structure. They use a different thing. Anyway, and later on they are going to, for example, they are going to use receptor and going to induce the IL-1 beta, which is another pro-inflammatory cytokine. Okay, a different pathway. The reason why our white cells need to equip different receptor, different function, and because activation of white cells is very important for our defense system. So they don't want to, they need to have different structure with the same mission. Is how to say that word? Uh, compensate the structure or mechanism. Okay, can overcome the defect. In case like a mutation, I still another set of stuff to overcome it. Okay, IgI like a receptor which particularly like mediates RNA. Okay, when virus infection, they release their RNA into cytoplasm, so they are going to recognize them using this receptor and turn on the gene. This gene particularly useful for interferon gamma. Okay, interferon gamma is used for attack viral infection, right? Okay, so I spent a lot of time go over the very important receptor on those neutrophils, macrophage, dendritic cells, which can recognize enemy and their stuff, right? And next part, innate immunity, including macrophage, of course, and the neutrophils, they also has another kind of weapon, phagocytosis here. They need the cytoskeleton protein, they need the pseudopodia, sometimes in public they need some kind of receptor. They internalize those pathogen or particles or chemicals into the cytoplasma, and those things was enwrapped in the lysosome. Okay, in this vesicle contain a lot of acid stuff, can degrade, can execute degradation or those substance. Okay, compared to the phagocytosis, there are another three kinds stuff. Okay, for the engulf the particles or pathogens or toxin substance. The first one they use Catherine mediate endocytosis, and another one the caveoli mediate use a different protein. Another one is a macropinocytes, which they don't need anything. They just use the actin film to increment. You gotta know one thing after this lecture. Phagocytosis store the particles into the lysosome, right? And the other type, non-phagocytic pathway, they store the the particle in the endosome. So it's not a lysosome. So endosome is a lysosome different. They are going to go over the ER, go over the stuff. Another organism. Lysosome will digest in the physical only. Okay, that's the difference. But you learn one thing, phagocytosis is another weapon for macrophage for neutrophils. Another thing you can memorize it, neutrophils after attack pathogen, after engulf the damaged cells or toxic substance, in the end, they are going to die through the apoptosis. Those apoptotic cells, the neutrophils, eventually will cleaned out by macrophage using phagocytosis pathway. Okay, so phagocytosis also engraft the leukocytes for reuse their material in the future. Okay, and also red cells also. Okay, now next slide, we are going to introduce three granulocytes, including basal, neutrophils, eosin. And here's information here is a little bit small to me regarding their size and their function. Okay, little information regarding basal. Okay, and the eosin probably maybe relate to allergy something. Okay, and let's look at the here is the neutrophils. I would like to say neutrophils. A lot of different granules, primary, secondary, tertiary granules, equipped with the different stuff, chemical in their granule, okay, including free radicals, including protease, including lipase, okay, a lot of different things which can kill a pathogen, okay, so it's very useful weapon for neutrophils. So you learn another weapon hiding in the granule or stored in the granule, phagocytosis. In the end, I'm going to say that in the end of life of neutrophils, it's going to undergoing apoptosis, right? Apoptosis is a cell death program. And neutrophils will use the rest of his material for defense. It will secrete DNA, RNA, and a histone protein, make it very sticky around the injury tissue. This sticky like a web, like a net, which can stop or slow down spreading or pathogen, which can block or inhibit the spreading of pathogen from the initial site to remote area. Okay, so the net, we call this process, we call netosis. So we know, so after that, we know a different weapon show in the neutrophils. Okay. Here is the letter I try to say is a neutrophil extracellular tract nets. Okay, you can see that they are going to die. They are released DNA and histone protein, right? And what happened? They will stop. They are going to. I'm sorry, it's here. They are going to stop a lot of passenger, make it sticky. Okay, and the other end, they are going to send a signal to the macrophage. I'm old. I'm dying. Please use me. And the macrophage will move to them and execute the phagocytosis and use their stuff again. Okay, and this is a very important step for cleaners injury site. Why? Because there's no left or debris or neutrophils in the damaged tissue, and it's good for healing, recover the tissue. Okay, improve the wound healing, accelerating wound healing. Okay, so we finish the parts mostly granular size, right? And then we are going to move to another cells in our bloodstream belong to innate immunity, monocytes. I told you activated monocytes are called macrophage, right? Okay, macrophage will turn into different subset of macrophage when they receive the TNF alpha or interferon gamma. Okay, they are going to become differentiate into the M1 macrophage. If they receive IL-4, IL-13, they are going to turn into another subset of leukocytes. We call it a macrophage, M2 macrophage. M1 macrophage is pro-inflammatory macrophage, which can kill, can clean the macro and neutrophils, can do phagocytosis, and benefit our immunity system or immunities and defense. Okay. However, M2 macrophage represent opposite function, which decrease or depress immune defense function. Okay, so they are totally different. So a lot of scientists working on cancer biology, they found a lot of M2 macrophage around the tumor and in the microenvironment, which can help, not kill the tumor, they help the tumor cells for progression, for migration. So a lot of study try to figure out what kind of mechanism or candidate which can block activation of M2 macrophage or reverse from M2 macrophage into M1, which kill the tumor cells. Okay, this is important topic so far in working on cancer research. Okay, next one. Uh, Another type of macrophage, we call them dendritic cells. You can see in the beginning of this lecture, I told you a different macrophage in our tissue and organ, right? For example, in our liver, there are special macrophage live there. They are coffer cells, which are very important for detoxification, right? When after the meal, a lot of stuff will from a portal vein going to the liver, a lot of toxic substance, for example, alcohol, right? Lipid microorganism will suck into will transport into the liver. Our coffer cells will, assess those toxic substance and ask you for phagocytosis and digestion. Okay, detoxification. That's the coffer cells. Microglia in our neurons protect the surveillance on our brain, CNS, right? Langerhans cells under our skin on epidermis cells. Okay, so many different specialized DC in our organs. Okay, if you have time, you can spend the time to look at it. Okay, actually, dendritic cells like a security guard in the tissue. Okay, when they find that the tissue got injured or enemies stay there, they are put into alarm signal. Okay, later on, I'm going to address this part. Okay, and later on, let's go over another different granular size. What's the type of individual? Okay, let's I try to say those security guards. I mean the dendritic cells or macrophage. They will. I told you right. They are going to use phagocytosis. They will. They will put alarm signal. What kind of alarm signaling? I'm going to inform my another. I mean the how to say the word? Say the word. Friend, army friend to try help me out for defense. Okay, I'm going to tell you that they are going to use energy presenting methodology. Okay, after they engulf the things, including virus, bacteria, right, and a drug, what happened? They are going to process it through the endosome or through the lysosome. They are chopping off the bacteria or toxin substance into very, very small things, peptides, sugar, or lipids. And eventually they are going to transport those fragments onto the receptor I just mentioned in the beginner class, MHC class 1 and MHC class 2. Okay, then present this energy and tell my friends, especially T cells, say I got to invade or I found the arm, I mean the pathogen around me, around my tissue. So you gotta come over here to support me, help me to fight it. Okay, so this kind of stuff present antigen from the foreign stuff onto MHC receptor. That's like a indication. So we're guiding these T cells, naive T cells, which is inactive from the T cells. Recognize this MHC class through the TCR receptor, okay? T lymphocyte receptor, and binding them, okay? And what happened? They are going to through another signaling, 80, 86, CD28, something like that, and then kill them, okay? Or help them to release different stuff. And another one, I'm telling you, there are two types MHC class, right? 1 and 2. What's the difference? After this lecture, you got to know that class 1 will bound to CD8 positive cells, T cells. Okay, it's kind of cytotoxic T cells, which will directly kill cells. Okay, why? Because class 1 represents the pathogen invading into cytoplasm. So it's very useful to kill the virus, okay, viral infection. It represent, I mean, the antigen represent or handled by MSC class 2. It means what? They use the phagocytosis pathway, engulf the particles and process them. So they tell you that CD8, CD4 T cells, you need to secrete a lot of signaling, growth factors, cytokine, a trigger, a different gradient to guiding neutrophils into the injury site. Okay, so that's the T help cells, CD4 T cells. So you got to know the different function. MHC class 1 for CD8, MHC class 2 for CD4. And CD8, I mean the CD8 T cells, which is cytotoxic T cells. CD4 is T helper cells, which is accessory T cells, which can secrete a lot chemical cytokine interleukin, which can amplify different type of leukocytes, including innate and adaptive T cells. Okay, so I'm going to tell one thing between the different type of immune cells during the inflammation. They use chemokine growth factor interleukin as the messenger that pass the signal, allow the cell to activation, allow the cells to inactivation. Okay, gotta know this concept. Any questions so far? If it's still confusing, please let me know right now. Okay, can I continue? If you can continue, please let me know. Okay, let's continue. I just mentioned that, right? Then DC macrophage B cells or professional antigen presenting cells. You can see this for APCS. You read the literature, you see these three words, APC, mean they can do and can present the antigen through their MSC receptor, okay? And recently they have found master cells, basal, eosin, ILC3, these kind of cells also has this function, okay, but it's not typical professional APCS, and there's some function on it, okay, but there is not that powerful compared to professional APCS. And just here, so MHC class 1, MHC class 2, both them are self markers, right? I told you in the beginning. Each cell has a unique sequence on their MHC class 1 and on MHC class 2. And these two protein, two receptor, and also responsible for present antigen to tell the to set the alarm, set the signaling to other immunocytes. Okay, got it? Here, viral infection, a lot of DNA or co-protein stay in the cytoplasm. They will process it and then present on the surface through the MHC. And this is MHC class 1. Why? Because these cells are invaded. They are sick. So they need to get rid of it. They don't want the virus reproduction in their cells. So what happened? CD8 cells come in and kill it, digest it, prevent their progeny production, okay, or new life cycle here. Other kind of cells, other kind of APC, they found a lot of different, I mean, the LPS, lipoprotein released from the bacteria or pathogen. They will do phagocytosis and get transport into the cytoplasm and process it and present on the antigen, either on MSC class 1 or MSC class 2. Two, if shown on the MSC class 2, it will attract CD4 passed into here and will secrete a lot of different cytokine, okay, and activate other type of cells, okay. If through MSC class 1, the same here like cytotoxic T cells will increase the gradient or numbers of CD8 T cells around the tissue. Either kill these cells or kill the other cells are infected, okay. That's the function of major histocompatibility complex or MHC protein. Is that clear? Understand it? Hopefully. And next slide here, I just mentioned in the beginning also, the aging or apoptotic neutrophils will engraft by macrophage. Let's look at this one. You know, apoptotic cells always will turn to the dense DNA in their nucleus. So you use the dye standard, you can see a lot of dark spots on their nucleus. Okay, and we want to. I mean, this is picture derived from the lung. Okay, lavage. Okay, alveolar lavage. You can see big cells here and a small cell here. You can see big cells inside a lot of dot. Very strong color here. It's suggesting macrophage engraft or eat those neutrophils already. That neutrophils already, apoptotic neutrophils. And then, I'm sorry, they put into electron microscopy and got macrophage neutrophils. So use this image, tell you, show you what the cleanness of neutrophils for the aging for apoptotic neutrophils by macrophage through phagocytosis. Okay, here's the, you know, this picture come from my former colleague. Okay, we are working on this project, a migration from bone marrow to, you know, to bloodstream. You can see that at night, neutrophils stay in the bone marrow. In the daytime, they move to blood. Okay, and at night they go back there. Okay, and when they get in age, you know, not normally we don't suffering inflammation, right? We don't get sick. So what happened? Neutrophils has a very average half-life, 24 hours, something like that. After that, they undergoing apoptosis, need to be clean. Okay, in where? Bone marrow. So they are going to travel back to the bone marrow, and over there, macrophage will found them, they eat them in the macrophage at night. Okay, that's how to maintain homeostasis of neutrophils, control the amount. Okay. Okay, in here, in the additional information, I mentioned that in the macrophage, they are going to release different stuff, free radical, nitric oxide, okay, and kill the cells, and also can what? Here's a monocyte activation, right, through the MSC and TCR receptor, okay, get activation. And also they can through the FC receptor. What is FC receptor? FC receptor, which can recognize antibody. I'm going to show you later on. The antibody is like a Y shape. They are viable region and constant region. Okay, viable region which can recognize antigen. Constant region we call it FC. So we and the macrophage can get IgG or antibodies. Okay, by phagocytosis. Okay, and the monocytes also very important for the wound healing. That's the reason why I tell you this immune system not only for defense but also for healing process is very important. Okay, because they have a different cytokine or growth factors. And the macrophage also has what? A function to bone resorption, which, you know, how to maintain a balance between the osteoblast and the osteoclast. Osteoblast is for bone forming cells. Osteoclast is the bone depleting cells. Okay, so they are going to make a homeostasis in the bone. Okay, that's the function of monocytes. NK cells, I told you already, is kind of like adaptive immune system, but actually they are innate immunity, which their function is they found cells are infected. As they found it, they are going to these different things which are also contain lytic granules. Release a different enzyme, performing and a granzymes. These two things which can make a pull on the cytoplasm. Okay, then what happened? These target cells were leaking their cytoplasm contents into the extracellular in my environment. And then eventually those cells were undergoing necrosis or apoptosis. Okay, that's the that's NK cells. And also it's very useful for defense tumor cells, okay, NK cells. So we finished innate immunity, right? Show you different weapon, different subcell leukocytes. Now we are going to go over another type of cells. We call it adaptive immunocytes, including B cells and T cells. I might go through too fast. It's okay. Okay, now we are going to look at these T lymphocytes here. You look at this picture, new mice. Have you performed any experiments on the new mice without a hair? No? Okay, good. In TMU, we use a lot of this kind of mice for doing drug testing. Drug is can be a therapeutic drug for treatment of cancer or not, different type of cancers, because we can inject our cancer cells from the human subcutaneously into the mice, into the new mice, and this tumor can grow. Think about it, the tumor cell line derived from a human, right? And this is a mice mirroring. So they are not going to get rejection. Allow the cells, allow the tumor can grow and turn to the mass here. The reason why those mice are depleting or dysfunction on thymus. Okay, we know thymus is a very important organ for maturing of the T cells. So this type of cell, this type of mice without the T cells. Think about it, without the T help cells, they are lacking a lot of different growth factor, cytokines, so their immunity is not perfect. Okay, it's immunodeficiency mice, which allow us to grow our tumor on animals. So we are observing the tumor growing onto around 500 millimeters square, then we can give a drug candidate. And we compare to control without any drug, we will see it's will evaluate, assess their therapeutic effect. Okay, use the new mice. Okay, so use this mice to tell you the T cells need to mature in the thymus. Without the thymus, we're impairing immune system. Of course, T cells originally derived from hematopoietic stem cells in the bone marrow, but they do need to move to thymus for maturation. Okay. However, thymus will degenerate with the age. Okay. For example, now, according to my age, my thumbs is getting very, very slow. So that's the reason why the aging of people has less functional or effective immunity in our body, because T cells getting number getting low and low. Okay, and this you can see that's the tumor. At the surgical removal, you can get this tumor here. We can image the volume and size. Okay, let's look at the T lymphocyte subset. T cells, they have unique marker CD3 positive cells and will present a different marker. T helper cells is CD4, right? And the CD8 will represent the CD8 positive cell and also interferon gamma here. And regular T cells, another set of regular T cells will present a CD3 positive, CD4 positive, CD25, and will release IL-10 TGF beta. I want to emphasize one thing: cells and cytotoxic T cells, it can benefit our immune defense against tumor, against the pathogen. Regular T cells, however, is suppressive function, inhibit the normal functional immunity. Okay, so but it is very helpful for patients suffering with autoimmune disease to lower down their immune function. So for those patients, medical doctor or scientists try to boosting the activity or number of regular T cells and decrease the function on cytotoxic T cells or T helper cells. Okay, that's strategy. Okay, so far for autoimmune disease. I'm going to ask you questions. You grow the T cell in the tissue culture, right? How can you tell this is T helper cells, this is cytotoxic T cell? What is a regular T cells? You will say by markers you should show on the slides. So how to determine those markers on the surface of those T cells? Have you know this machine, flow cytometry? Are they using this in your school? Flow cytometry, which is an instrument, allow you to tell subset of leukocyte or cancer cells combined with the dye, fluorescent dye. Okay, for example, I got these T cells, right? This type of cells, maybe a different compound, maybe T helper, TCD8. It's a mixture over there. I don't know how many percentage of TCD4, CD8. So what happened? I'm going to use antibody against CD4, CD3. CD3 are both share receptor. CD8, they are different, only expressed on cytotoxic T cells. CD25 only regular T cells. So I'm going to use different cocktail antibody conjugated with different dye, fluorescent dye. Okay, so this cytotoxic T cell will live after applying no antibody. They were recognized by CD3, CD8, and this one will recognized by CD4, CD3, and this was CD3, CD4, CD25, and were reported by fluorescent dye. Okay, and those cells will go through a hole on the instrument. Only allow one cells one time. Okay, then we are going to give the light energy, and those fluorescent dye will absorb it, right? And it will turn to a different wavelength for release energy. And those machines will accept those kind of changes, and it will report it. These first cells, when they pass through this hole, they show these two kind of color. And this one, they show different color. So we can simultaneously to recording everything. Okay, then later on they will show you what's the percentage, what type of cells, and also in the end they got a sorting. They only collect these two markers. Another chamber collect these two markers. Another chamber collect 3 marker positive cells. So use this methodology to isolate and to distinguish different type of cells from cocktail cells, from the mixture cells. Okay, flow cytometry. If you got a time, you can go to the YouTube or go to Google, try to find out what is flow cytometry. It's very useful to study immunology, okay, or bone or stem cells. Okay, that's strong suggestion to have this kind of expertise in the future. It's good for you. Okay, now we let's look at CD4 T cells here. This is very important chart here. Here's antigen presenting cells, and he showed the antigen presenter MHC class 2, not okay. Then MC class 2 were recognized by TCR receptor and also were recognized by CD4 to affirmation, to confirmation. After the activation, what happened? These CD4 T helper cells will start to release, different in the cytokine group and the cytokine, chemokine, and growth factor. For example, interferon gamma is very important for viral infection, right? And T helper cell will release IL-12, interferon gamma, and cause T cell to turn to Th1. And if this T cell secrete TGF beta, will allow this FOXP3 and stage 5 positive cell convert into Treg cells and release different other immunosuppression factor or cytokines like IL-10, TL35, TGF beta, something like that. Okay, that's the reason why these APC active T helper cells can modulate different type of T cells and with their effect. So depleting T helper cells will cause immunodeficiency disease. Okay, the most case which we know that, let me see, okay, it's HIV virus. HIV virus with the receptor love to connect or bind to and the CD4 receptor, especially the GP120. After recognize them, they will internalize. And what happened? Their genetic back information will integrate into the DNA or host. So their viral DNA will stay in the host DNA. So it's, in other words, there are no drug efficient to kill this virus because they hide in a host already. And eventually they are going to amplify a lot of baby virus. Eventually we're depleting CD4 positive cells, T helper cells. So we're killing off all the CD4 T helper cells. So what happened? Cause immunodeficiency in the patient. So you can see this kind of sarcoma cancer appear in the patient. That's the sign. Kaposi sarcoma is a sign for patient, for individual develop immunodeficiency disease like AIDS. Okay. So T helper cells is very, very important for their activity. Next one is CD8 positive cells. CD8 positive cells, as I mentioned, was activated by antigen presenting cells through the MHC class 1. Okay, and after the activation, they are going to release. They are going to recognize cells got infected here, for example, and through their TCR, and will release interferon gamma, IL-7, and some kind of protease enzyme, which these two enzymes we mentioned already, will punch the cell membrane into the hole, okay, and change the osmotic pressure here, and leading to cell death, infected cell death. Besides that, the activated CD8 T cells will develop into cytotoxic T cell 1, cytotoxic T cell 2, cytotoxic T cell 17. Okay, 1 and 2, you can see that 1 with a cytotoxic, activity. And TC17 is appears in mucosa with the activity, with the immunity, with the strengthened immunity. But a TCL, TC2, which one is suppressive function, okay, in active immunity, I mean the function here. So there are three types of T cells. Okay, here this picture is very important for you guys. For example, you got a vaccine, right, for COVID-19. You need a 2 shot. The reason why, because B cells need to 2 activation events to get into mature plasma cells and produce a lot of antibody into the shedding into bloodstream. Okay, for example, this one, when it, for example, we got the vaccine, right, with the inactive form of virus, for example, or particles of virus into our body, and that into our muscle, right? There are a lot of Langerhans cell living there. They will receive those kind of antigen. They will, they are APC cell. They are going to present this antigen onto the where their MHC protein, right? And the dendritic cells will migrate into the lymph nodes. Okay, because a lot of T cells and B cells living around the lymph in the lymph nodes. So dendritic cells meet those T cells and B cells in the lymphoid nodes and activate them. And T helper cells can improve the maturation of cytotoxic T cells and also improve B cells, B cells. So eventually, these naive B cells will turn into the plasma cells, but it is short lifespan. Okay, they will squeeze antibody, but not enough. The amount is not enough for our immunity for vaccination. So after probably 2 months or 12 weeks later, or I don't know, 8 weeks later, we are going to give the second shot for boosting our immune system. So what happened? Again, dendritic cells will pass the signaling to the B cells and the BC cells. Eventually, they are going to turn to the plasma cells. This type of plasma cells, again, is mature and long-lived, can secrete a lot of antibodies, IgG. Okay, initially they maybe produce IgM, but this time a lot of IgG already. So they will achieve to protect COVID-19 infection because a lot of antibody in our bloodstream will neutralize by the particles. Okay, so after this one, you know that the reason why you needed 2 shots of vaccine. Okay, and they tell you the reason why. For example, as use for example, baby. I remember when my child at the 10 months old, I got a visit from actually my mentor with their kids. They coughing. They were coughing at that time. After the meal, they left, and next day, my son developed a flu syndrome, coughing, high fever. Okay, because he was a 10 months old baby infant. He relied on the antibodies from the mother, right? Left over from the milk or bloodstream. At that time, his B cells his B cell never exposed to those type of antigen. So he was suffering high fever, very severe syndrome. With the time passed, when getting old, those plasma cells encountered same antigen couple times already. So next time when a virus infects, or I mean the pathogen infects, he can respond very, very well because he encountered those antigen not only twice, a couple times already. So he has immune to those kind of energy. Okay, but for the baby, probably it's new to them, so it's not make good enough antibody to against those pathogens. Okay, that's the reason why we encourage the mother for young mother to how the breastfeeding their baby, because there are a lot of IgE in there, can help benefit baby's immunity. Okay. Next one, the IgG plasma cells, mature plasma cell will release IgG immunoglobulin. The structure immunoglobulin like this, you can have a full chain, two light chain, two heavy chain, okay, like a white later, okay, and also you can see two different color. I mean, two different region. This region is, I mean, the antigen binding site, okay, on the top, and this part we call it FAB. This is a very important region to recognize antigen. Another part here is FC, FC region, and also here it show you the FC region has more constant region. I mean, the sequence is not changed that much, but a variable region in the energy binding site here is changed a lot. Okay, between the light chain and heavy chain, and between heavy chain and heavy chain using disulfide binding. Okay, make a four peptide together to form IgG. Here is the structure domain. Okay, what's the function of FC? Can bound to the FC receptor. Appear on the receptor. It show up here. Okay, it's kind of protein, right? From this from the C3, you can see C3 where the C3, C1, C2, C anyway. Eventually, we'll make a C3 and make a hole, punch the hole, and make a cell death. Okay, this kind of protein, and so you can imagine that FC receptor and the IgG can bound to the antigen, bacteria, or toxin substance over there, and then later on complement will bind to another site, and also will recognized by macrophage. So what happened? Will assist them being phagocytosis, being eaten by macrophage. Okay, you gotta know that the function, complement, and the immunoglobulins. How many isoform of immunoglobulin here? IgG, IgE, IgD, these three very similar structure, like a letter Y, okay? IgM is pentamer, which is contain five IgG linked together, okay, through this linkage. And okay, IgA actually is dimer. Okay, you can see there's different in structure. Okay, IgG is mostly useful for neutralization, for binding antigen. Okay, and there are two different subclass: IgG1, IgG2, IgG3, IgG4. That depends their FC region. IgM, which okay, is a compound appear on the plasma cells. So plasma cells show up as IgM, which when they inject a vaccine, right, the first shot, they put in IgM, not the IgG. Okay, you can remember that everything first the encounter some antigen will induce IgM from the plasma cells, not IgG. I mean, most the case. So you look at the curve. IgM will increase a lot, and later on will induce IgG. Okay, and IgA expressed appear to be a dimer. Okay, it will exist in the mucous tissue. Okay, and for secretion. And IgE involve the allergy. Okay, and IgD function is still unclear, not clear. Okay, IgA probably a lot of probably appear in the milk also. Okay, so you gotta know the differences between those different forms of antibodies. Here, what's after this lecture, you're gonna know what kind of cells, what kind of cytokines or growth cytokine or chemokine involved in this process. I'm gonna show the one example. When a lot of students, I mean, foreign students traveling to the United States and then go to the national park. Okay, there are a lot of beautiful trees, and also during the springtime, they are sent. I mean, a lot of pollen was shedding off from the tree, right? So when we walk into the state park or national park, we encounter pollen, right? So what happened? Antigen presenting cells will process the fragment of pollen and show and then present on the MSC class 2 and activate our T cells. Eventually will activate our plasma cells into mature plasma cells. Okay, but it's short-lived. But some B cells will develop memory B cells and memorize this kind of fragments, okay, or peptide. Second year 2, we went to a state park again. Okay, then we encounter second time for this pollen. Those memory B cells will react very aggressively and produce a lot of IgE. Okay, those IgE will stick into master cells and recognize pollen. And pollen will happen, activate those master cells. We know master cells can secrete a lot of histamine and making our runny nose, and coughing, and fever. Okay, that's the reason cause the allergy through the master cells, through the IgE. Okay, gotta know this one. So that's some people. If you you go to another different place, you live a second year, no pollen disease. It suggests that you probably have a little chance to get an allergy for pollen. Maybe, I don't know. But sometimes people get suffering after 4 or 5 years later. Okay. Now we are going to go over complement, which is mentioned about that. I wish all you know this after this lecture. What's a complements? They start, okay, there are 3 different pathways here. Okay, I told you they are protein, right? Struct protein or protease released from the liver, produced from liver. What happened? The first one is alternative, the classical way. This one is what? The bound to Fc receptor start from the C1 from the complex one. Okay, eventually they are going to turn to the C3 convertase formation. Okay, and another one is an alternative pathway, which they don't need to recognize Fc receptor. The hydrolysis in the cytoplasm turn into this pathway. Another one is lectin. Okay, lectin, which is shedding from the bacteria or other kind of stuff, or dead cells, okay, or aging cells, which can activate the complements also. Okay, their function will increase a membrane attack complex, which is thread the hole I mentioned already, and also increase chemotaxis effect, anaphylaxis. Okay, this is a very acute allergy. Okay, so complements has this kind of function. Okay, you can see that. Another you can see that, for example, that's a macrophage, right? and the C3b, this fragment will bind to my antibody here. This is antibody. Will recognize antibody. Antibody recognize my micro B. The antigen. So those cover a lot of complements will help macrophage to eat them. Okay, so that will assist macrocytosis. Okay, that's the first function here. And second one again, they are to what? Recognize complements label on the micro. The passaging here. What happened? They will recognize by neutrophils or macrophage here. Then will release their toxic substance and degrading those antigen. Okay. Okay. And number three, I mentioned already, I make a hole here and cell eventually will functions of complements. Okay. After that, we have another system. I think I gotta go do very fast. Lymphatic system here, including the different type of structure, lympho, which is a body fluid, okay, and a lymph vessel, which connects to another lymph node, okay. And also has a bone marrow and the spleen, okay. Spleen can produce a lot of lymphocytes during the fetal life, okay. But after bone, the adult or baby, they don't. Only bone marrow can produce the lymphocytes. Okay, and they got several functions. They can transport excess body flow back to the bloodstream. We know that lymph nodes can drain a lot of body fluid into the lymphoid system, right? And if there are too much lymph in the lymphatic system, they are going to move some liquids into the bloodstream and maintain a balance, okay? Maintain the pressure. Another one, protecting body against disease causing. What happened? Because you see this structure, they a lymph vessel doesn't directly connect with the blood, even artery or vein, okay? But they got functions. If they are harmful toxic substance or tumor cells or pathogen in the blood, eventually they will go to the lymph nodes. Lymph nodes with the function as a filter to start with those cancer cells and the antigen or toxin substance. And in there, T cells and macrophage will eat that, will digest. So you act as a filters. So will protect our body, prevent the loss of pathogen or tumor cells and migrate into distant organs or tissue. Okay. Another one is very important, absorption of fat from the small intestine. Okay, this is very important subject also. So that's the basic function of lymphatic system. Okay, here the lymph node. You can see a lot of naive T cell and T cell, B cell here, and also dendritic cells were moving in after the antigen presenting cells. In the lymph node, they will contact with the T cells or B cell activated. Eventually make a lot of cytotoxic T cells, make a lot of plasma, mature plasma B cell and shedding antibody. Okay, for complement the immunity. Okay, here I'm going to show you a disease, a abnormal growing or lymph nodes or the lymphatic system. We call them lymphangioma, infangioma. Okay, and this kid actually he's my son, and when he was 30 years old, probably third grade, 10 years old, he suffering the abdominal pain, severe abdominal pain, and then we go to the medical doctor and find out he got a mass on his peritoneal tissue. Okay, then after the surgery, spent probably 10. I forgot how many hours, 10 or 6 hours, a long time. Then and the doctor in the end, the doctor showed me this mass to me. You can see almost 20 centimeter small intestine here, and the tumor mass. And this kind of is born with that. Okay, when he was the baby, he was the in his mother's. Um, uh, when he was how to say fetus, he developed this kind of okay, this lymph node. Okay, and actually he's very lucky. He's easy to surgically remove this mass, but for these two big kids, it's much tougher, difficult on the neck, on the tongue, and some kids develop on the brain. So it is very difficult to do because a lot of neurons, a lot of venue over there. Okay, so it's not that easy in this case. So that's another kind of disease. Okay, now we are going to explain what's the inflammation, acute inflammation. How many people know this? I'm going to demonstrate that. I'm going to show you as point my view of inflammation. Okay, for example, unfortunately, I make a cut on my skin on my fingers, right? I'm started bleeding. My tissue got damaged. So this will generate a lot of signaling. I got the damage. Okay, I release tissue factor, cytokine, and shed the signaling into the cells surrounding the injury site. For example, Langerhans cell, dendritic cells here, and the master cells. Okay, and what happened? Master cells will release histamine. Histamine can increase blood flow rate. Okay, increase flow speed, increase vascular tone, increase the gap between endothelial cells, right? And the blood will, we are bleeding, okay? So increased blood flow, a lot of neutrophils and the macrophage monocyte will infuse or infiltrate into the injured site, okay? Then what happened? The dendritic cells is first the person that recognize damage of tissue is going to eat those damaged tissue into fragments, or they found a invaded passenger. They are going to execute the phagocytosis, and then after process those potential antigen, then will turn into the MHC class 2 or class 1. Okay, probably MHC class 2. So we'll show the signaling. I got it invaded, I got it damaged. So those dendritic cells, as I mentioned in previous slide, they are going to migrate into a new lymph nodes, activate the T cells and B cells. T cells will turn to T helper cells and cytotoxic T cells. Cytotoxic T cell will migrate into the injury site to kill the infected cell in case if you are lung injury, if you are COVID-19. And the B cells will shedding a lot of plasma cells. Okay, and shedding a lot of antibody from plasma cells. And the neutrophils will go in there, do the clean up job, okay, clean everything because he has a lot of number of the neutrophils. And then release different weapon, free radical, or lipase, okay, protease, something like that, okay, try to kill a passenger around, living around there, or eat their metabolized stuff, toxin substance. Or eventually, after a couple of days later, they are going to doing the healing process. Okay, eventually those neutrophils getting old, aging. They are going to undergoing apoptosis, as you mentioned about it. The macrophage will clean up those neutrophils and get a balance. Eventually, our wound is healed. Okay, nothing happened. The whole thing we call it inflammation. Okay, and have any idea now? Okay, you got inflammation. So one question is that how initially how those white cells can mobilize going to injury site, right? You need a lot of neutrophils. How can you shed in a lot of army into war zone? Very easy. When it got a cut, I told you they was give some kind of signal, mechanical signal, chemokine, which can attract the neutrophils, can activate neutrophils. Chemokine can activate the neutrophils. The endothelial cells, putting the expression of adhesion molecules, okay, on the endothelial cells, on the white cells. Those molecules, including selectin molecules, okay, including integrin molecules. Selectin molecules, for example, L-selectin, P-selectin, and E-selectin. L-selectin is very useful for lymphocyte migration in the lymph node. That's the reason why they label with the L lymphocytes. P-selectin expressed on platelets and also endothelial cells. E-selectin was secreted from the endothelial cells. Okay, when you get an injury, neutrophils need to move very, very fast. The first contact adhesion molecule is L-selectin. After activation, L-selectin will shed in onto the bloodstream. And later on, probably 10 minutes, P-selectin are stored in the granule. So they don't need to the process of activation, the transcription label. They have a P-selectin in the granule. So in the 10 or 20 minutes, they are shedding on the endothelial cells. And our neutrophils has some kind of receptor will recognize the E-selectin. Okay, so they are going to move right now, very fast pattern. 3 hours later, because our injury will induce secretion or produce a lot of TNF-alpha, something like that, that kind of cytokine, will activate E-selectin suppression. E-selectin is much smaller receptor adhesion molecules with higher affinity to bound to neutrophils or macrophage or monocyte. So neutrophils will slow down in the the site near to the injury site. So they can initially they put a lot of cell in a faster pattern and slow down in the injury sites. So at that time, we'll aggregate a lot of neutrophils or monocyte in the damaged tissue. Okay, so in a very short period of time, we recruit them. Then eventually, another molecule we call an integrin express in integrin for firm adhesion and help those neutrophils or monocyte cross over endothelial cells here directly or from the gap on endothelial cells and migrate into the tissue. In the tissue, they were doing the cleanup job, as mentioned in the couple slides before. Okay, now this pattern we call them very important. Leukocyte adhesion cascade. A lot of pharmaceutical companies developed drug to block those events, signaling adhesion molecules to minimize the damage autoimmune disease, okay, or inflammation. Okay, I'm going to show you
Prof,
how scientists.
I'm sorry, Prof.
Yes.
Our class time is over.
Can I? It's not to 1 o'clock. I'm sorry. Can I give me some more minutes?
Yes.
Okay, thank you. Thank you for reminding me. Okay, I will go fast. So scientists use this methodology to determine what kind of adhesion molecules are important for leukocyte adhesion cascade. For example, we use intravitreal microscopy, which has a large stage, can put the animal over there. Okay, and we put the buffer in there because we do surgery, the tissue will dry up without the buffer, right? So put in there, and we need to keep the buffer with a 37 degree, otherwise the muscle will twitching. So we cannot get a good image. So with those mice, we do that. We need to put them sleep for more than 12 hours, long time. So I'm going to use a sleeping drug with uracil and correlates, and mouse will put sleep. Unfortunately, will develop mucus on the trachea. So we try to breathe. So we are going to do tracheotomy to help them breathe. Okay, after that, mouse is going to happy, right? So in this case, we are going to watch the leukocyte loading in the tissue in the venue. So we are going to find a muscle which is easy, allow us to easy or image those vessels. We found one, the vasculature in the muscular, muscular muscle, which the muscle in grab surrounding the testicles. Okay, so this muscle only appear minimized. Okay, here at the surgery, you put the stage and it we spread out the muscle and see a lot of vasculature. Okay, and then put into a microscope in the TMU and then we can do image, we can do filming in the real time. Okay, and then we can see, for example, like this one. And this is another model, sickle cell disease model, because this setting is highly inflammation. So you see a lot of leukocytes is loading fast pattern or slow pattern, something is sticky over there. Okay, so use this methodology, we'll check. This one is, if I'm a good surgeon, there's no activation of leukocytes, no activation of adhesion molecules. So it's very clean. After 10 minutes later, leukocyte loading very, very fast. In this stage, leukocytes mediated by P-selectin, which facilitate fast loader. 3 hours later, we see leukocyte loading slower. Okay, then eventually can turn to firm adhesion and go and found the target site and then migrate into the tissue to do the cleanup job. Okay, if you knock out P-selectin and E-selectin, you can see that there's no activation of neutrophils or monocytes. It's clean, like a good surgeon after surgery. So I use this methodology to tell you how to approach to understanding those adhesion molecules. Okay, for example, and it is similar to the flow cytometry, tell you how to determine the subset of leukocytes. Okay, that's the methodology. Okay, here I'm very quick. I told you, right? During inflammation, the T cell will get activated, will secrete a lot of interleukin 23. Interleukin 23 will help out activate IL-17 T lymphocytes, secrete IL-17. IL-17 will upregulate GCSF. GCSF is the mobilization agent, can mobilize stem cells from bone marrow to bloodstream, can mobilize neutrophils and macrophage. So a lot of macrophage will go into the bloodstream, go to the tissue. Okay, after clean up the damaged place, neutrophils is dead, right? And there's no signaling for macrophage. IL-23 drop, IL-17 drop, GCSF drop. So no more neutrophils mobilization from the bone marrow to the bloodstream. So we turn off this loop. Okay, so no more neutrophils. For a lot of autoimmune disease, they cannot regulate IL-23, IL-17. So a lot of neutrophils come into the damaged tissue, keep release a lot of toxic substance for your healthy tissue. So we're turning into autoimmune disease. Okay. For example, this kind of disease, a lot of neutrophils, macrophage release a lot of cytokine in your finger. What happened? They eat their bone, right? And a lot of things. You see that's the reason. Okay, that's autoimmune disease. And also, I'm using this figure to tell you that I told you immunity is very important for defense passage, defense cancer development, right? Progression. But however, I told you a lot of different, the immunocytes will develop into different sets of subset of leukocytes. For example, macrophage, T1 to T2, M1 to M2. M2 is not good for us, it's good for tumor. Okay, neutrophils also neutral tumor-associated neutrophils also can help the tumor to progress. Okay, to progression to meta. So a lot of people working on this field around the tumor microenvironment to double check what kind of tumor cell, what kind of immune cells good for us or bad for us. Okay, so it's a new topic for cancer biology. And here is a whole set of white cells. Okay, they derive from the bone marrow. Okay, the 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 reason why we know those important stem cell differentiate into different subset of leukocyte white cell platelets because this event during the end before the end of World War II, to atomic bomb dropped into Japan, right? A lot of people died months later because infection. So people make people curious how which area is produced immunocytes, white cells or antibodies. So they use a different method. They use the late to cover the major organs and put the animal into the chamber with the X-ray or something like that or radiation. They eventually found out if those protection from the bone marrow, they can protect the mouse die at the sublethal elevation. That's the reason why they found bone marrow is a site for progenitor cells or stem cells over there. Okay, so after we know that they are the stem cell over there, they have two major events. The first one, what is the first one? Degeneration. They can produce identical layer cell, lens cell. Another one, they can do into multiple lineage. Or for example, blood cell, T cell, B cell, myeloid lineage. Okay, and the first one, MPP, is myeloid progenitor cells. Everyone got to listen to it. What's the difference between stem cells and progenitor cells? The same thing is they got the capability to differentiate into different lineage, different type of cells. But stem cells is capable to do regeneration. They can produce themselves renewable. Progenitor cell cannot make them own. Okay, they just has the capability to differentiate into different lineage. There's a difference between stem cells and the progenitor cells. Okay, so in my lab we found out, not my lab. Okay, we found in the patient after treatment, after chemotherapeutic drug, they found a lot of stem cells in the bloodstream. It's suggesting that stem cells were mobilized from bone marrow to the bloodstream. Okay, this observation is very helpful for bone marrow transplantation. Okay, in the past, we use this big needle, okay, get into the bone marrow. It's painful, it's risky. So there's not much people want to be a donor, right? For harvest the bone marrow for people suffering leukemias or chemotherapy. So right now they are very easy. For example, in my lab, we found a lot of agents which can increase mobilization of stem cells from bone marrow out the bone marrow into the bloodstream. Okay, so what happened? Right now we just sit into the on the bed. Okay, just like a blood transfusion. Okay, all the cells go into the machine. They will figure out just I just mentioned, if side by markers. Stem cell has their unique markers. We store those stem cells with their markers, then sit over 5 hours, 10 hours. They collect a lot of stem cells and then they give the recipient. So we don't need to do the penetration into the bone marrow. This act, okay, that's very good for. So I would use this figure to tell you that a lot of scientists working on this project, how to increase mobilization of bone marrow, how to increase inflammation stem cell into bone marrow for visual differentiation. Okay, that's immune. Okay. Next one, in the end of couple slides, this one is Nobel Prize winner, both to share on the physiology and medicine in 2018, because they found very important stuff is immunotherapy. Those two proteins is immunocheckpoint. Edison found the CDL of A4, and the IP and the PDL1 was PDL1 was found by Japanese Honjo, Professor Honjo. Okay, these kind of inhibitors which are with the inhibitory function, inhibit the T cell activation. Okay, tumor cells will produce a PD-L1. PD-L1 can bind to the PD-1, which inactivates, suppress the activation. So right now, a lot of drug pharmaceutical companies, they develop PD-L1 inhibitors, block the negative control. Okay, so it's a very hot topic right now. Okay, so I share with you. If you interested in this topic, go to online, check PD-L1, PD-1. Okay, I think in the next 10 years, maybe a lot of drug will try to develop immune checkpoint inhibitors. Okay, in the end, the picture I took in China is Grand Wall. If you have a chance, you should go over there. It's unbelievable memory over there. Okay, this wall was built on the nowhere to go. I think here is on a couple of mountain. Okay, thank you. Every time I spend provided too many slides, but I just try to share my knowledge or what I learned to you guys. Okay, thank you. If any question, please let me know. Thank you very much.
Thank you, Prof.
Okay. Any questions?
Prof, maybe one question in the short time. I want to ask about the COVID-19 vaccine. Health workers in Indonesia get a third dose of vaccination for a booster. What is the meaning of a booster? Does it has relation with the work of Selby? Prof, thank you.
Can you repeat again? My signal is not that great. Can you pardon, please?
In Indonesia,
yes,
health workers get a third dose of vaccination for a booster. What is the meaning of a booster? Does it has relation with the work of Selby?
Boost, you mean this 2 shot or what? Or another thing?
3 shot.
3 shot.
3 shot.
Okay, it's difficult question to me because there's no evidence to show 3 shots is a better second shot. Because most, you know what I'm saying?
Yes, yes.
Yeah, I don't know, because so a lot of scientists, they encourage the United States or European countries, you don't need to do that. Second shot is good enough already. You save your vaccine for a country or for people. A lot of people did not see any vaccine yet. Okay, so it's a debated issue, though, I think. But if you can get a third shot, that's fine. Okay, but I don't think it will work. And the second 2 shot, there's one topic you can do the research. I suggest you can check how many people get 2 shots already still develop severe syndrome, COVID-19 syndrome. If not, that's fine because you got a protection already. Right now, we are going to look the data regarding infected. Okay, we are looking for the data, severe disease syndrome need to hospitalize. That's the most important issue. We got a flu, we don't go to hospital, right? We take a rest 7 days. That's the mission, that's the goal for vaccination, for protection. There's no way to diffuse virus. You cannot come after get a 3 shot. No way. You still get infected. But a syndrome is minor. That's good. Okay, that's my opinion. Okay, I don't encourage people to get a 3rd shot. Okay, it's not fair. But you need to at the least 2 shots can get complete protection for your health. Everyone got a 2 shot already in your country? Well, that's good. Congratulations. I still had one. I still got one shot already. A lot of students, it's still waiting. Because my country, they don't have the plan to import vaccine or vaccination. That's bad. Because we did pretty good job using a mask. Okay, the incidence is low, but look at the how about a couple months later, we are not going to travel overseas. You know, it's not good. Okay. Any questions?
Okay, thank you for the answer.
Thank you.
Any other question, class? No more? Okay.
Did you have
Elizabeth?
any questions? Any
Elizabeth?
comment?
No?
No.
They say no, Prof.
And I speak too fast, too complicated.
Can you catch class? Can you get
Any
what,
idea?
Prof? Okay. I think they are starving, Prof.
Start to think. Okay, you can go back. I'm going to upload the slide. Okay, go back to see
Okay.
the second page. What's the content? You can know. You can say what's innate, what's adaptive, what's inflammation, and what the components. You can recall those things. You are good now. Okay.
Yes, Prof. So maybe if they have question, they can contact you in your email address like that, Prof.
Sure,
Is it
no
okay?
problem. I love to. Okay.
Okay. Okay. If no more question, I think we will close this class today, Prof.
Okay. Thank you.
Thank you very much, Prof,
Thank you
for
very much.
your time.
Okay.
Okay, good
Okay.
afternoon.
Okay. Bye bye.
Thank you, class.
Thank you,
Bye
Prof.
bye.
Thank you, ma'am.
Okay.
Bye. Bye.
Bye-bye.
Bye bye.
Thank you, Prof. Thank you, ma'am.
Bye.